The session title is Neurodegeneration, Causes, Treatments, and Societal Impact. Our moderator is Carol Manning. She's just walking up there. And our panelists are Ishan Williams, nursing, Mimi Riley from law, John Lukens, neuroscience, and Heather Ferris, endocrinologist, School of Medicine. Take it away, Carol.
Great, thank you. Well, I am really excited to be here, and I have to tell you, I have a great group. I have been so lucky to work with them. But I think you've probably noticed every group has been great, and in addition to that, the themes have been really similar across groups. For those of you who've been here all day, you've probably noticed that whether we're dealing with autism or early development or Alzheimer's disease, the themes are the same. And the session that we just heard was really fantastic in terms of helping us understand all of these issues. So, I'm going to do something a little unusual here. I would like you to raise your hand if you know someone or have ever known someone with Alzheimer's disease. Okay, look around you. I hate to say it, but thank you very much. That is basically everybody in the room. Even when I talk to undergraduates and I ask them that question, the answer is the same. Even young people, almost everybody knows someone with Alzheimer's disease. Unfortunately, the current state of the treatment for the disease is such that if things continue the way they are, either everyone in this room will get Alzheimer's disease or will be caring for someone with Alzheimer's disease. So we have a big problem that we need to solve. There are currently, I meant to introduce myself. I'm Carol Manning. I'm a neuropsychologist. I am the director of the Memory Disorders Clinic in the Department of Neurology in the School of Medicine.
And I'm Mimi Riley. I have a primary appointment in the law school, but I also have appointments in public health sciences, Batten, and the Miller Center.
Good afternoon. My name is Ishan Williams. I'm an associate professor in the School of Nursing, and I'm a social scientist.
Hi, my name is Heather Ferris, and I have a primary appointment in the Division of Endocrinology and Metabolism, but I also have a joint appointment in neuroscience.
So I'm John Lukens. I'm an associate professor in neuroscience. Just from some of the earlier talks today, I was thinking, why am I so nervous today? And I realized the last time I was allowed to speak in the rotunda was my public defense for my PhD at UVA. So I'm pretty sure those circuits are pretty strong.
So the good news, John, is I'm assuming you passed, right? So we're okay. So back to our subject here, which is dementia and Alzheimer's disease. There are currently 6 million people who have Alzheimer's disease. The trajectory of the disease is really long, on average 6 to 10 years. You all said you know someone with Alzheimer's disease. It starts out as something that you may notice mildly in terms of slight memory impairment, but as the disease continues across the years, people lose even their basic activities of daily living. They no longer remember who their loved ones are, they can no longer brush their teeth, bathe, do even our very basic things that make us humans. Caring for someone with Alzheimer's disease is intensive and it grows over time, and we're going to talk a lot about that. The costs of the disease are enormous. In 2021, the costs are projected to be $355 billion. Given that age is an enormous risk factor for Alzheimer's disease, as our population ages, this is going to completely do in our Medicare system. There is no cure. Unfortunately, we have no cure for the disease, despite there being over 300 failed trials. And I can tell you that at UVA, we have participated in more than a few of the clinical trials, and it is hugely disappointing, certainly to us, but more so to the people with dementia and their caregivers, that we do not have a cure for the disease. Heather, can you talk a little bit about why you think we do not have a cure? What's gotten in the way?
Sure. So, the approach that we've been taking up until now for treating Alzheimer's disease is waiting until a patient has symptoms. And we know at that stage that there have already been many neurons lost. Neurons, unfortunately, don't grow back like so much of the rest of our body. And so once those neurons are gone, all we can do is hope to augment what's left behind. And that's really too late in the course of the disease. So we really need to start looking earlier, targeting people before they have symptoms. And that, I think, is where the hope is for preventing Alzheimer's disease. Not so much treating it, but preventing it.
And John, do you want to say a little bit about the people who have been in studies? Why?
Yeah, so I'll talk a little bit about where we've been looking at Alzheimer's disease patients. And most of the time we've been looking at familial patients. So these are people that have defined genetic mutations that make them at risk, and they develop Alzheimer's disease at a very, very early age. So if you've ever watched 60 Minutes or read the New York Times, you probably know of this community down in Colombia where there's really high rates of Alzheimer's disease. And these are genetic forms. And this is what the field has been focused on, looking at these, what they call early onset or familial AD cases. Unfortunately, these aren't the types of Alzheimer's disease that most of our loved ones are going to face as they progress in age. And the other forms are more idiopathic and sporadic, and we don't really necessarily know exactly what's involved. What we do know is there's multiple genes involved, and that there's also environmental factors, things like poor sleep, poor diet, bad periodontal care. There's all these environmental factors that are coming to light that are linked to Alzheimer's disease, and we think that our plan could help better understand what mechanistically is underlying those.
So now we're going to talk a little bit about the other sufferers in the disease, and those are, that is the caregivers. There are currently, our current system really relies on unpaid caregivers. That is typically a spouse who is often the same age or older than the person with dementia, or sometimes a child. The care that these unpaid caregivers provide is 16.6 billion hours of care a year at $244 billion. That is enormous, and that is in lost income. So caregivers, not only are they providing care for their loved ones, but they experience increased morbidity, higher mortality, and they have lots of biomarkers of poorer health when we compare them to age-matched controls. They experience more stress, more depression, more anxiety. They have overall poorer health. And really of importance is they are at increased risk for dementia themselves. They are at 6 times increased risk for dementia themselves than age-matched people who are not caregivers. And so if you think about what Heather and John just told us, that's incredibly important because we have a group here who we know are at increased risk for the disease, but they're not showing symptoms of the disease. So they provide this perfect model or this perfect group for whom we know that a lot of them are going to get the disease, and we have them before they are showing signs of the disease. So there's a model here for people at risk who we can study. Based on that, we want to talk about how we can have a very novel model of studying Alzheimer's disease and caregivers, which will provide a model where we can be comprehensive and holistic, which is really the only way we're going to move the needle in this disease. What we're going to talk about is utilizing an existing area of expertise, which is caregiver science, which some of us have been doing up here for quite a while. And through that, we are going to improve the health of our caregivers, ease the burden, change policy. You're going to hear more about that around caregiving for patients with dementia, both in Virginia and across the entire country. But we also can move the needle in terms of understanding the onset of the disease itself through looking at caregivers. One other thing we can do is, John was talking about the current treatments, if you will, that we have. The best treatments that we have are behavioral interventions, including increased exercise, improving diet, increasing cognitive stimulation and social interaction. And these are all things that we can implement via the caregiver to the patient. So we can actually change the trajectory of the disease in the patient through the caregiver. Well, one of the ways that we're going to do this is through creating a clinical cohort, which we will talk about. But we're going to do this first by talking about our areas of expertise. And let me start by talking about the clinic. I am the director of the Memory Disorders Clinic. And the Memory Disorders Clinic here at UVA is a multidisciplinary clinic. We are lucky to have neurology, neuropsychology, physical therapy, occupational therapy, speech and language, social work. We have a pharmacist, and we early on learned that the identified patient, that is the person with the illness, is not the only one who's suffering. So we treat the family, we treat the patient and the caregiver. Our clinic has won national awards for provision of care, and we have funded research on the impact of caregiving, including increasing knowledge of dementia, providing support, and managing the disease more effectively. So we have already in place a way to utilize our clinic to further research on patients and caregivers. So I'm going to, John, would you talk to us a little bit about the research that you're doing that can play into this?
Yeah. So in case you didn't notice, I'm an immunologist. I'm not really a neuroscientist. Don't tell my trainees that. They already know that. But so I guess the one thing I wanted to talk about, and I've kind of switched what I'm going to say right now from earlier, is just based upon some of the questions that people have been asking. And like, okay, what are examples of grand challenges that have worked in the past? What have we learned already, and how do we apply that? And I think back to when I started my graduate training here at UVA. There was this idea of cancer immunology. Some people might know these individuals: Vic Engelhardt, Craig Slingoff, Tim Bullock, and they were saying, okay, we're going to harness the immune system to fight cancer. And we're like, okay, Vic, just teach us T-cell development. And you would go to these meetings, immunology meetings, and there'd be very few people at the cancer immunology talks. And you go to cancer meetings, and there'd be very few people at the cancer immunology talks. And I remember the discovery of the molecule that now is a drug happened 20 years ago. It was a Hanjo paper. It was an immunity. They just won the Nobel Prize for it. But that wasn't too long ago. It really wasn't. And it's just been amazing how quickly that field has evolved into something where everybody was like, okay, this is going to be a fad. It will be here and gone. But we're here today, and it's changing people's lives. I mean, if you know of anybody that has had cancer or who's suffering from cancer, they're going to take these drugs. This has changed the way that we treat patients. It works really well for things like lung cancer and some skin cancers, basically outside the brain. So when thinking back 5-10 years ago, there were very few people talking about neuroimmunology and the ability to harness the immune system to treat neurological diseases. One of the people that many of us have always looked at crazy was Yoni Kipnis, and we still look at him crazy. But that was a unique opportunity to come here to UVA to do something different. And there's a huge opportunity to target these molecules that have been identified and that are now well received. I mean, we're in the midst of Society for Neuroscience. That's the big meeting for neuroscience. And just going through the program yesterday, if you compare that 5 years back, how much neuroimmunology is there, it's just mind-blowing. And one of the major drugs that people are targeting now is a molecule called TREM2. And this is a very important molecule that can kind of boost the ability of microglia to remove toxic compounds from your brain. That discovery of that gene was 7 years ago. It was only 7 years ago, and now there's phase 1 clinical trials on that. And the one thing that I'm hopeful for, and what pushes me to come to work every day and excites me is the thought that someday soon there will be people that are treated at UVA across the world with immunomodulatory treatments to treat neurological disorders. And unfortunately, in the past, we've really focused a lot on the neurons. And there really aren't any effective treatments for things like Parkinson's, ALS, Alzheimer's disease, some tauopathies. And there's a tremendous opportunity to change people's lives. And I think, I'm new to the caregivers model, and it immediately made sense to me, because where you study your biology is so important. And we've been looking at these rare populations for so long. It's time to switch things up. And I think this is a really unique population. And I was really excited to be part of this exercise.
Heather.
All right, thanks, John. That was a great introduction to really, I think, what all of us have felt about this caregiver model and how unique it is as a doorway into prevention. I'm an endocrinologist, so I'm a little bit of an oddball on the panel, but I got here because patients with diabetes have a twofold increased risk for dementia. And so, just like Carolyn's been using caregivers as a lens for trying to understand the underlying mechanisms of disease, we've been, in my lab, trying to understand the mechanisms through that lens of diabetes. And it really translates very well. I take care of patients with dementia in my clinic, and I take care of a lot of the caregivers, and those caregivers have horrible cardiovascular disease, they have hard to control diabetes. Some of this is because of the stress of the caregiving, some of it is because they don't have the time to invest in their own care because they're so split between themselves and the person they're caring for. And so how can we come up with models that ease their burden and then try and understand how that changes physiology in the patient and in the caregiver, and then how that translates down to molecules that we could hopefully one day convert into therapies. Whether it be that we're modulating the immune system, maybe not through a drug, maybe through a behavior change even, which would be wonderful in the accessibility of it. So I think that this is an opportunity to hit the problem very broadly from many different angles.
Okay, all right. Well, thank you, John and Heather. As I mentioned in my introduction, I'm a social scientist. My background is actually human development and family studies, and I'm a family gerontologist. So I came to the School of Nursing to really focus on my research in looking at culturally appropriate interventions for family caregivers from underrepresented populations and those groups that are disproportionately affected by Alzheimer's disease. And I also, linking to what Heather just said, I also link that to populations like the Black populations are disproportionately burdened with Alzheimer's disease because they are disproportionately have type 2 diabetes, cardiovascular disease, high blood pressure, you name it, where have the health disparities that exist in those populations put them at even a more increased risk of having Alzheimer's disease. And so some of the work that I have been doing with family caregivers really has shown that what we need to address in these behavioral interventions, not just looking at patients and how patients get the disease, but how family caregivers looking at the stress and the burden that is put on these family caregivers and how they themselves can be part of the intervention to reduce the negative health effects and the negative emotional effects of being a caregiver. And as Carol has identified, we have seen the evidence showing that caregivers are going to be at risk for getting Alzheimer's disease. And so part of my research has been to really focus on not only doing community-engaged research, but community-based research. So actually taking some of this clinical stuff that we're talking about here and finding out how to put that in community settings to get this bi-directional feedback where the community can they themselves share with us their needs, their relevancy of the types of projects and research that we're doing so that it can then influence the way the scientists are utilizing the research questions and the types of products and outcomes that they develop so that it is relevant for all populations. We're trying to be as inclusive as possible. So if I had a separate moonshot for the community, it really would be to make sure that all of our diverse, like all of our clinical trials, all of our research projects that we do have diverse representation from underrepresented populations as well as rural groups geographically, because as Charlottesville is like this one city surrounded by rural populations, and so we have an opportunity across the state of Virginia to really make an impact in this area, being culturally and community-based strategies.
So I like to think, Ishan, that I'm coming back to you. Don't worry, I haven't forgotten you, Mimi, but I like to think that your moonshot, your mini moonshot is a part of our big moonshot, right? Mimi, have not forgotten
And
you.
actually, what I would say is my role is largely to take Ishan's community moonshot and take it to the country.
Yeah.
And you probably noticed by my affiliations, I'm kind of multidisciplinary, except I don't know anything that any of you know. So I'm doing it from that world. But I do view part of my role is as an interpreter, taking what they are studying, what they're learning, what they're doing in the clinic, and then taking it both into the public at large, but also to our policymakers. As we think about Alzheimer's disease, the economic impact is unbelievable. It is going to mean that there are so many things we can't spend money on. Education, for example. Other types of issues that we're trying to bring forward as a society. And so we have to think about that. So many of the issues that we're dealing with here are issues of policy. Many of you in this room have been paying attention, as I have, to Aduhelm. We're going to keep coming back to this. The idea of how you approve a drug with fairly limited data, how you use an abbreviated pathway, these are ethical decisions and actually need to involve the community. One of the reasons that we have when we think about Aduhelm and its approval was, well, people are desperate, they need something. Well, before you make that assumption, you really need to address the caregivers and the patients and think about what the best way of dealing with these issues are. We really haven't studied that much. I've been working with NIH on some of these issues where we're engaging the public and asking some of these questions as we think about emerging biotechnologies. Another place, even in the research side, we need to engage the public. So many of the tools that we are going to use as we think about dealing with Alzheimer's involve organoids, embryoids, which actually have major ethical concerns for the public, and we need to start having the public understand why the science is going in the direction it is, what those tools are going to teach us, how we can do them better, and we need the dynamic of the public as researchers telling us how that should be done. Similarly, for example, primates. I've been working at the NIH with primates as a model for neurodegenerative disease. I can't tell you how controversial it is. We have to go in secret rooms as we're working on these issues. But those are issues where, again, we need to engage the questions and take some of the policy questions. All of us had a connection to Alzheimer's. That's a connection we need to build. Last point, you talked about Medicare and Medicaid being bankrupted. They will be, but part of the reason is what we pay for. We don't pay for behavioral interventions. We actually do our reimbursement in a completely sporadic and ad hoc fashion. We need to engage the public and the caregivers using these dyads to think about how to make that more effective and get value for your payment.
So, Mimi, most people know about Aduhelm, but would you just say a word to describe what happened with that so people are aware?
So Aduhelm, and I could probably spend a class because I have, but very briefly, to say that Aduhelm was approved through a recognized abbreviated pathway with, I would say, controversial data. Most of the studies on Aduhelm had failed. One study actually did not fail. It had some statistically relevant data, and on that basis, Aduhelm was approved. The difficulty is, one, it's tremendously expensive. It's $50,000 for a course. And the other thing, it has major side effects, one of which came out in the news today. There is a patient who died, and Biogen is actually studying to see whether that was related to the drug. And we're grasping at a straw when we actually need to actually think about the disease in a holistic manner in such a way that rather than just throw things at it, like Aduhelm, I think we could think about these behavioral.
And I'd like to say we were one of the trial sites for Aduhelm, and that we actually had 2 patients who experienced that cerebral edema, that brain swelling. It's no joke, it's really serious. And so when people make the argument there's nothing else, actually the data that they found they culled the data. It was a subset of the data that they then reanalyzed, and the results were no better or no more impressive than exercise or diet change. Thank you. You can see here we have an amazing panel of people that go from basic science to policy. That is UVA. That is what we have here. We are really lucky. And we have the ability to move the needle. So our moonshot idea, you can see, is to identify and implement effective interventions both to treat and prevent neurodegeneration by studying partner caregiver dyads. We can study people, partners and their caregivers together in tandem. This is a strategy that can utilize all of the talents you see here, plus everybody in the room who you've already heard. So what we're going to do is having a clinical core where we're going to recruit patient caregiver dyads. And these patient caregiver dyads are going to come in together. They're going to have an initial visit and then annual visits, where at each visit we get biomarkers including neuroimaging, cerebrospinal fluid, sleep measurement, cognition, mood, look at healthcare utilization, so we can go on up to policy, and look at these people over time. This clinical cohort is going to be available to everybody here, anybody at UVA who is interested in any kind of research involving neurodegeneration. This is a cohort that is going to be absolutely unique across the United States. Ishan, do you mind saying a word about our the success we've had in recruiting cohorts?
We have a lot. No. Well, I can't say enough about our success in recruiting without saying that it comes with a lot of effort and attention, being very intentional about the way that we engage our community partners, which is actually really responsive to the National Plan for Alzheimer's Act, the NAPA Act, where they are really hoping for institutions and organizations to build collaborative community partnerships. And we can actually maximize the work that we do in community settings through our own federally funded CTSA, our iThrive Center, that helps us keep costs down, because For someone who does a lot of recruitment with underserved populations, it takes a lot of time, and it takes a lot of money, and it takes a lot of building trust and relationships in these communities. So you need actual human capacity to be able to do that. And we'll talk a little bit later about infrastructure and who we can have to bring on our teams to help us with recruitment. But we've been able to really successfully recruit rural populations across the state of Virginia to come into clinical trials. Myself and Carol Manning have been really successful at recruiting African American and Black individuals over the age of 60 who have vascular risk disease. So we know that people are interested in doing clinical trials and research studies. It's often that they're not asked or they don't have the knowledge and understanding. So that's where this community engagement and outreach area of excellence, I feel like we have a lot of people, including those on the panel today, who are really committed to making sure that we have diverse representation in all of our samples. One of the critiques about the Agilum study is that they had less than 1% of people of color in their sample across, like, over all the sites that we had, right? So it's really important that we talk and have knowledge, help communities understand the importance of research, what that means for them, and then how they can be our partner. So this education and engagement piece is where not only do we go to the community and provide education about how to do research, what this research really means. When you talk about neuroimaging, what is that? Because most people just don't understand what that looks like. But also having community come here to UVA to actually have this, allow there to be a lot of feedback. Because often I hear from clinicians, it's like, wow, I forgot what it was like to have an actual human person in front of me, you know, like talking to me, because they're in the lab so much working on these other things with animals or with other types of items. I don't know what, I'm not a, you know, yeah. I work with people, so I always want people involved. But again, you know, thinking about how we can break down the walls of UVA to really make sure that our community is getting the needs, their needs met. Because if we're going to be an innovative leader in Alzheimer's disease, we cannot ignore the disproportionate communities that surround us. And I think that's really the importance of the community engagement outreach.
Heather, would you talk some about the data sciences and biorepository?
Sure. So Carol just outlined all of these visits and all of this data. We're going to have amazing access to clinical samples that we don't have right now. And so we're going to need to create a biorepository, and then we're going to really need to engage the members of our data science community to be able to mine this data, to connect it to the electronic medical record, to make discoveries based on that data. And so I think that we're really well positioned with the growth of our data science department here. And this really, as Carol said, unique resource. This cohort doesn't exist anywhere else. And it will be something that I think a lot of us will want to use and hopefully we'll be able to share with others around the country as well.
John, can you speak to mechanisms of disease, please?
Yeah, so a key component of our Moonshot is going to be this reciprocal crosstalk between the basics and clinical science, where breakthroughs that are made in the lab are taken to the clinic and incorporated in the care of individuals with Alzheimer's disease and also their caregivers. And we're going to, in a couple minutes, a couple slides, we're going to get into some examples of how we think this could work, both in terms of like the basic experiments, how we can take it from bench to bedside, and then also what kind of impact this would have in terms of community outreach and public policy and the good aspects of how our Moonshot works. But in addition to the ones that we're going to talk about, there's plenty of other opportunities that we think would be key to kind of jump in on. And one opportunity would be to look at lymphatic drainage, as was mentioned earlier. And we know that lymphatic drainage leads to this buildup of neurotoxic material in the brain. And it would be really cool to see when the caregivers develop this kind of block in this drainage, and if that's a legitimate biomarker for early onset. And then taking the basic component of that, there's a bunch of us that are working on compounds and novel ways to boost this drainage pathway, and seeing if we can incorporate those when the drainage does become impaired. And there are various other examples that, as we go longer in this, we could talk about.
And Mimi, do you want to talk about social sciences, law, and policy, please?
So I could talk all day, but I'll just focus on a few things. One is when we start talking about data science repositories, we're talking about privacy issues, especially when you're talking about issues that involve Alzheimer's and the development of Alzheimer's. The other thing is when you have caregiver patient dyads, they may not always have the same interests at all the time. So you have to think about this as you're designing both your cohort and the studies that may be involved with those cohorts. And so we would have that put in place from the very beginning. So there would be a constant dialogue between these societal issues and some of the research issues that we'd be looking at the same time. On the other side, thinking about some of the policy things, think about, most of us have been thinking, or I have anyway, about the infrastructure bill that has caused so much political mayhem in Congress. Part of that infrastructure bill, the better care, better jobs piece, if you look at it, is a significant portion of money for home and community-based services. Wouldn't it be nice if that was science-based? If we actually thought as a social science and biomedical science question, what services are actually needed? And so what the idea with these cohorts is to actually have a dynamic interchange going back and forth between end policy and research questions and back to the policy questions. So you don't actually have it done, this sounds good, but rather think about it as a comprehensive system-based network theory-focused policy.
So you've done a great job describing some of the ways this can work, but to give another example, Heather, would you talk about a possible sleep?
Sure. So a lot of you here are parents, and I'm sure you remember that first 6 months of your child's life and how little sleep you got and how really terrible it was. But it got better as time went on, and you had something that was really great that you were looking forward to, and you were young. So try doing that as a 70-year-old who's watching your spouse get worse and worse and worse, and think about how this is happening over the course of years. And so this sleep disruption is really so incredibly hard on the body. We know that it increases stress hormones, it probably changes lymphatic drainage in the brain and the ability to clear toxins from the brain. There's so many different aspects of physiology that this is potentially hitting on. And so one idea would be, how could we introduce an intervention with the caregiver, patient dyad, maybe home health aides who could relieve the caregiver for a couple of nights a week so they could get good sleep. And then we could look at the patients and how they do, but also probably more importantly, we can look at the caregivers. And are we changing markers? Are we improving the brain lymphatics? And so come up with rational ways to intervene and benefit the caregivers. But then we're also getting all of this biologic data from the patients that we can take to the bench and find specific therapies. Some of the things that John's talking about, looking at can we introduce a compound and mimic the same behavior? And so it has a huge potential to benefit those caregivers, but really we know sleep is a really early sign of Alzheimer's disease, and so maybe then we take it to the patients early on and see if it could help prevent disease in the patients themselves as well.
Thank you.
All right, so metrics of success. So we wanted to talk about a couple of things that we thought would be important as far as knowing that what we're doing is making a difference. And You know, for the example that I just gave you, it's really finding those interventions that have a biological outcome that we can see that's changed. And we see all sorts of R01s and research grants coming out of that, where we're really exploring the mechanisms and taking it from the bench to the patient and back again. But long term, really, we aspire to change the course of the disease. To find things that are going to long term prevent disease in patients and move the needle on this really debilitating process.
I think some additional short-term outcomes that we can look at, to your example, Heather, is going to the community to really address some of the social determinants of health that we know create barriers to how we develop programs to address sleep, to address exercise, that we know that when caregivers are stressed, it disrupts their sleep, and then that makes it limited amounts of exercise that they can get. So how do we work with the community to develop programs that would really reduce their access, right? Not improve their access, but provide community programs that addresses those issues around sleep and around exercise. And we know about diet. I don't know if John is going to talk more about diet or not, but those are programs that we can make sure that are relevant to the communities that we really want to serve. And that's easy to take the expertise that comes from these innovative ideas and these projects to the community. Those are short term to me. Long term, I think we can look at the diverse representation across all of our projects and works that we produce out of UVA to see that we are tracking to make sure that we're having the investment that we are committed to across all of the studies that we want to see. And these are things that I think the National Institutes of Health are interested in helping us to reduce health inequities across all of these diseases, but more importantly, in these communities where there's disproportionate burden.
Okay. John, did you want to say anything? Nope, you're done? Mimi.
Well, I was going to count on John to talk about. So, John, talk about what you're going to talk about.
You want to talk about diet and microbiome?
Diet and
Someone?
microbiome.
Oh, yeah. So
I need to channel that.
we skipped the slide, so, but it's okay.
Sorry about that.
Yeah, so one of the ideas that we have is, okay, why are caregivers so prone to being at risk for developing neurodegenerative disease later? And they're the perfect incubators to acquire the microbiome that their loved ones have. So we know that Alzheimer's disease patients have altered microbiomes, and the microbiomes are the trillions of microbes that live in us, and they're involved in almost every disease. And we've kind of missed them because we can't culture a lot of these bugs, but now genetics, we know that they're highly responsive to things like stress and lack of sleep, and diet. And this could be one way in which the caregivers become more prone to developing Alzheimer's disease or another form of neurodegenerative disease later in life. And diet is a big component of that, and that's one way that we can kind of stabilize the microbiome. And you know, there are a lot of communities out there where there's these food deserts, and there isn't access to great food, and it comes even worse when you're in this stressful condition, when you're caring for loved ones, and you're just trying to get food in your body so you can keep going. And so as a way to improve the community, we could think of creative ways to bring education to the fact of how important diet is for your neurological health and how it can help prevent you from having dementia and all these neurological conditions later in life. And that was one idea that kind of came out of our exercise, and I'm sorry I forgot it.
Yeah. And to take John's science and show how policy plays a role with just very low-hanging fruit. We know, for example, that the microbiome in the mouth actually is associated with Alzheimer's disease. So if we look at that and we start studying and find causation, because that's always valuable to go from association to causation, but let's imagine we do. We have thousands of Virginians without access to decent dental care, with actually really not that much money. By intervening early, we then prevent all the costs we would have associated with Alzheimer's disease later on. Not all, some.
So
no one ever sell.
You've talked about some really practical things that we can do that are really low-hanging fruit to change the trajectory of the disease. So I know our time is up, but I hope that we've convinced you that we have a wide range of talent here at UVA, that we could bring in more talent to even grow that. NIH has let us know that they are very interested in the caregiver dyad strategy. And by getting P30 Alzheimer's Disease Center status, we know that would bring in lots of additional NIH funding. So this is something that not only would play on our current strengths, it would grow it and lead to significant additional funding. And then finally, this really fits in with UVA's great and good. It's great in terms of adding to the science of neurodegeneration, and it's actually good in terms of serving the community around us. So, thank you. Questions?
I really appreciated John's comments on the crosstalk between basic scientists and clinicians and how that relationship can inform some of the approaches to this topic. And so I wanted to bring to this conversation the sort of perspective of a researcher like me who is also a clinician in the peri-operative setting. I am an anesthesiologist by training, and I am a critical care physician. And so in the operating room, we are seeing increasingly elderly patients that are coming for complex surgeries. And oftentimes they come more than once. And so I take care of them in the OR, and then I also take care of them in the ICU. And what we're seeing is that oftentimes those patients who come in with pre-existing forms of dementia, oftentimes not even diagnosed Alzheimer's, but also other forms, are at hugely increased risk of developing what we call post-operative cognitive dysfunction, which is this whole spectrum that goes from acute behavioral changes like delirium in the ICU all the way to, other cognitive deficits that can be quite prolonged. And while we thought in the past that these were acute changes, it turns out actually that research is showing that some of these patients will never go back to baseline and actually will settle to a new baseline that's lower than prior. And then when they'll come back the second time for their anesthesia and surgery, which they will because they're not growing any younger, they'll be even at higher risk of developing these cognitive dysfunctions. And this has huge repercussions in terms of increased length of hospital stay, increased morbidity and mortality. These patients don't go home. We have to send them to long-term care facilities. So huge healthcare costs. And so in the field of anesthesiology and critical care and in the perioperative arena, this is a really huge topic. There's a lot of NIH funding, and I look at it as a very crosstalk area. Just thinking in the clinical setting, specialties like anesthesiology, internal medicine, neurology, geriatrics, surgery. These would be all disciplines that have an interest in this, and this is even without mentioning all the basic science that you guys even have mentioned in part. Sleep disturbances are a factor here. Microglia inflammation from earlier panels plays a role in certainly ICU delirium and post-op cognitive dysfunction. So just thinking that there's a lot of cross-pollination that could be going on here, and that this would be maybe a little bit different way of looking at this topic.
I can tell you, we see those patients in clinic every week. There is at least somebody who is post-anesthesia, and it could be a long while post-anesthesia and have ongoing cognitive decline. George. Oh, sorry.
Oh, so I actually had a simple question about the dyad model. And so do you plan to actually collect lymphocytes or peripheral lymphocytes and store them sequentially in the caregiver versus the...
So that was one of the things that came up in our exercises, is tracking these caregivers over time. And you know, it's easy enough to track how their immune system changed and how their microbiome changes. And then we're actually planning, Carol and I, over the last couple of weeks, to start taking microbiome from patients and putting them into germ-free mice with the help of TUMI, which is the great microbiome initiative that was just started 2 years ago. And putting them into germ-free animals to say, like, okay, this is the microbe, and then, you know, this is maybe the effect that it's having on the microglia or whatnot. I think one thing that's we could really benefit from at UVA is somebody that's looking at the CSF. So this is all the fluid that kind of cushions our brains. And it provides a kind of a window into what's going on in the brain. Because, you know, imaging is great, but when you want to get down to like a molecular, like what kinds of things are coming out of the brain and what's going on there, that really gives you a nice window into what's going on. And we're seeing more and more of this in the Alzheimer's disease field, of using this CSF, this fluid, as to track disease and early onset. And that's where some of the hottest biomarkers are coming up.
About 10 years ago, people refused lumbar punctures. They did not want to give us their cerebrospinal fluid, and it's becoming very routine, and that most patients who are coming through now actually get, in terms of the diagnosis, get a lumbar puncture. So we are collecting CSF.
Yeah, that would be the major goal. And you know, you could do single cell sequencing or mass cytometry. There's a bunch of groups here, Eli Zunder, Colleen McNamara. They're doing these longitudinal studies where they can actually freeze down the cells and stain them later with mass cytometry, which provides, you know, incredible depth at the protein level.
Hi, I'm George Bloom from biology. Many of you know me as running a sort of cellular molecular research lab about Alzheimer's disease, and I've already learned a tremendous amount about the other side, and I'm really excited about this dyad of patients and caregivers. And I'm astounded by the incidence, the high incidence of Alzheimer's in the caregivers. And while it may be entirely explicable by the stress that's associated with the caregiving and the effects that stress has on the immune system, it occurred to me that maybe there's an infectivity component to this as well. So there's at least one class of neurodegenerative disorders that's well known for being highly infectious, mad cow disease and all the related syndromes in other species in humans. It's basically the same disease, but mostly called Creutzfeldt-Jakob disease. It's highly infectious. It's not caused by exchange of nucleic acids, but rather of misfolded toxic proteins that build up in the patient, in one patient, and then are transferred somehow to somebody else. And then these toxic misfolded proteins direct the production of similar toxic misfolded proteins in the recipient individual. We now know there's a lot like that goes on in Alzheimer's disease, at least 2, maybe 3 proteins involved in this. And I wonder if you plan to look at that possibility as a contributing factor in this high incidence of disease in caregivers.
So, George, the beauty of this design is it is there for you as a researcher to look at exactly that. So,
I was hoping you'd say that.
Yeah.
Yeah, I mean, Toomey just put in a really amazing IRB that allows great flexibility. So you can take basically fecal samples from anybody, any disorder at UVA. It's an amazing resource, and you can do these types of studies by, you could first show it in an animal model and then extrapolate out what that actual bug is.
Along those lines, people come to UVA in part because they want access to research, and so people who are in these dyads, they will want to participate in other research beyond just the annual visits.
So a natural thing to add is amyloid PET, to do it in the dyads,
Right.
time synced, to see whether or not the progression, of mild cognitive impairments ultimately, you actually get a buildup of amyloid that tracks, but time delayed, that of the affected individual. And I think here you would have a biomarker, whether this actually is really related to and maybe an infectious disease, prion disease. I mean, that would actually then trigger the accumulation, the misfolding of amyloids.
Absolutely, and caregivers provide a great group to look at.
It turns out that it's something else. We have these repositories, and we can go back and say, all right, these were the progressors, and maybe we couldn't find something from the microbiome, but we still have these samples, and we can compare those that were progressors and that weren't, and find new avenues to go down.
One of the things that I'm incredibly struck by is the common themes across the day. And so, as you were speaking, thinking of being a caregiver of a child with autism and the caregivers we meet, all of the same issues. Even though we're younger, still that lack of sleep ages us quickly. When we look at our parents, there's higher rates of Alzheimer's and Parkinsonian. In the patients with autism themselves, higher rates of Parkinsonian. I wonder if, and thinking about cerebral spinal fluid, one of the biomarkers that's now replicated 4 times, which is really a miracle in biomarker research, that predicts which infants will go on to develop autism, is benign increased cerebral spinal fluid. So it's not something that would really get you flagged, but just something that a med student who was involved in research at 4 different, at a university that was part of our network ended up noticing, having just been through the class and asked about it, and we all started looking, so we're seeing this commonality. And that brings in this kind of what the Glio group was presenting earlier. I mean, it really speaks to the need for not just recruiting an Alzheimer's caregiver dyad or a NICU parent dyad or a baby parent dyad, but really a generational cohort, because of course that opens us up to being able to do whole genome sequencing studies and identifying what's rare, what's common, what's inherited, what's de novo, and really be able to look at across generations of a family and follow them over time, because all of these issues are kind of coming together, and that could, I don't know of any place that is thinking in that way, because again, the silos of disorders.
Completely novel approach, yeah.
You brought up an interesting point about the cerebral spinal fluid and the pressure that it puts. There's actually a trainee from UVA that just went off to Cleveland Clinic to start his own lab, and he's studying this now, and he's showing that there's actually impairments in the drainage pathways in many of these autism models, and it's really fascinating just to connect this to something that was mentioned earlier too.
So, a very quick question. So it's very exciting that. Okay, it's very exciting that we are collecting all this data from the caregivers. So I just want to know, what kind of privacy protections we have for collecting the data and analyzing the data.
So people sign consents before they can participate, and then we de-identify the data.
Okay. I think I won't take more time, but I definitely be very curious to know what kind of the anonymization of the identification you have done to the data, because that's lots of time probably it's hard to remove all the sensitive features from the data, even we remove some of the metadata. Yeah.
So the process that we use, we go through the IRB, obviously, and there can be no data that can be identified. With genetic data, there are different processes, but nothing that is identifiable about the person is kept with the data. So every individual gets a number, and the number is not associated with the name in any way other than kept under lock and key. And it's a standard procedure that we use all the time that when we are collecting human data.
Thank you.
But I think this brings up an important point around ethics, and again, educating the community about what data is being collected at the university across these many studies. And I think that is something that, I'm not going to say it's innovative, but we don't do enough of it, about what people sign, when they sign these forms, what are they actually signing their consent to do? And I don't think that, I think that's an education that can be done at the community level as well.
Especially, Ishan, since the consent forms, since I started doing this, they were 2 pages. They're now about 14 or 15 pages, and really hard to understand.
Right.
Most of which is meaningless.
Right, exactly. Yes.
Hi. I actually don't have a question just for this panel, but probably for everyone in the room, and Ram may be able to address it. From everything that I've heard today, from neurodevelopment, from autism, through imaging, through you, speaks about that the biggest moonshot in science overall is translation of basic research to clinical research, going to the community, and then going back. And what I would like to ask is, for all of us in the room, how can we really, truly address that at UVA? So what can UVA do to have us make that happen? Thank you.
Is that to the group? Or is
Or
that
the provost.
George always has the answer.
George has the answer.
It's not always the right answer, but we're doing it right now, and it's a consequence of the intense communication between clinicians and basic scientists. So a couple of years ago, a grad student in my lab discovered that a drug that's approved by the FDA for treating late-course Alzheimer's disease, mainly because in some but not all patients it provides very modest temporary relief of symptoms, but doesn't do anything at that stage to interrupt the processes that are leading to neurodegeneration. The student, Erin Kotis, she now works at University of Pennsylvania, discovered that this drug may indeed have disease-modifying properties, that it might be able to prevent or at least delay the onset of symptoms and slow the progression. It's kind of an indirect long story. But as a result of that, I've been communicating with Jadip and Carol and other clinicians here. We went through all of the IRB applications and everything, and now we're approved for running a pilot phase 2 clinical trial to test this drug as a prophylactic for Alzheimer's disease. So I think that demonstrates that we already have all the infrastructure, the human infrastructure that we need here to accomplish exactly that.
We are very lucky in the degree of collegiality that we have here. It's pretty amazing relative to other places. And as I look around the room, there are several people basic scientists who I am already working with doing translational research, which is really exciting. Okay, so I'm getting drinks. What are we doing now? Thank you, everybody.
All good things come to an end, but I want to say this is just the beginning of the conversations that will happen at UVA. What an exciting day. I want to give a special round of applause for all the panelists for their hard work. Please join me. We have heard exciting ideas, several moonshot questions, as well as this common thread needs. And also this interaction between panels, Adema, the question you asked, what does it take to do this? These are the questions we will contemplate in the coming days. And there are things that we can do, and there are things as a community you can do. I think this convergence is important. In fact, I used the word convergence. That's exactly what all of you talked about, that all the way from basic sciences to translational elements to bedside or molecules to minds. In any which way you frame it, you're actually bringing these communities. A variation on George's answer in that, if I paraphrase that, this is bidirectional. I think you mentioned about bidirectional flow, simultaneously designing these research projects. The clinician informing the basic scientist and vice versa in the dynamic environment is a step closer to what sits well with the UVA as our culture of collaboration. So thank you all for doing such a wonderful job. And special thanks to Liz and Jim for investing to grow research at UVA. And in fact, I should single out Liz here for clearing her calendar to be here all afternoon. I know how busy you are, how triple booked, quadruple booked you are, and I so appreciate, and all of us appreciate. A round of applause for Liz. Lastly, I want to say that the investment in research at UVA is unprecedented, especially in the recent past. And in fact, John Lukens referred to the TUMI. That's an example of an investment that's enabling great studies that didn't exist 2 years ago. There are many examples like that. So personally, as a researcher myself and speaking for all of us, it's an exciting time to be here at UVA and pursuing your dreams and research. So what a great time to be here. So with that, thank you for being here. Please join us in the reception at the multipurpose room. Go all the way down. When you get out the door, turn right and walk along the walkway, and I think it's the second door past your right turn. So, and have a good evening, and we hope these collaborations and relationships you have built will continue, and we'll do our part, and we will together move research. I have no doubt we'll move the needle towards preeminence. Thank you.